Hello, everyone. First of all, I would like to thank the organizer of this conference to give me the opportunity to present the QURGEN's work. And QURGEN is a protein-based regenerative medicine. And we have three technology platforms, and which are supported by strong independent intellectual properties. Our first technology platform is a protein delivery technology platform. And the second technology platform is a protein-induced iPS technology. And third one is a protein-induced in vivo tissue reprogram technology. So next three slides, I will explain each of the technology. And the first is the protein delivery technology. And this is a technology which we have is based on a series QQ reagent. And the QQ reagent is a QURGEN invention, which is a polymer-based protein delivery reagent. So we have a pre-made QQ reagent, and the protein could be any source. For example, could be bacteria expressed or mammalian cell expressed, or isolated protein. So all you need to do is just mix those two pre-made QQ reagent with protein and with a certain procedure, then we will have a QQ modified protein. At the in vivo cell settings, cell culture setting, all you need to do is just add the protein, QQ modified protein into cell culture medium, and then the protein can efficiently penetrate inside of the cell. So one of the key feature of this protein delivery technology is the very high protein delivery efficiency and safe. And so it could reach to greater than 97% of the protein delivery efficiency. And so not only that, and this protein delivery technology also has the intracellular specific intracellular organelle targeting capability. For example, I have an example here, which is a nuclear localized protein. When the protein was delivered inside the cell, then the protein will penetrate inside of the nucleus. And so this is an in vitro application. For the in vivo application, here we have a mice which has a brain cancer. So in order to perform early detection of the brain cancer, and we would like to use MR imaging to do that. In order to achieve this goal, and we identify a protein called ferritin, which is an iron bonding protein. The iron serves as a negative contrasting reagent for MR imaging. So what we did is QQ modified the ferritin, then perform a Tavern injection, and at a very low dose, which is approximately 20 to 50 microgram per mice. And then we just wait for 4 to 6 hours. Then we put the animal into sleep and perform the MR imaging. As you can see here, this is a QQ modified ferritin, and all the dark area are the brain cancer. You can see very nicely, and with a QQ modified ferritin, brain cancer can be mapped very clearly. But with ferritin injection, you don't see anything in the brain cancer. So this really indicating by a systemic injection of a QQ modified ferritin, and the ferritin was able to target into the brain cancer tissue. So how the protein delivery happened? So here is an example. We have a protein called OCT4, which is stem cell marker of a pluripotent marker, and we use a green fluorescence fluoroform to label the OCT4, then QQ modified. Then we add the 3 bag per ml concentration into the cell culture medium. In 10 minutes, you can see a very nice protein on the cell membrane. 20 minutes, the protein will penetrate inside the cell, got into the cytosol. 30 minutes, you can see those protein actually leave the cell membrane and reach to a very close to a paranuclear space. And the OCT4 is a transcription factor, which is a nuclear localized protein. As you can see, 2 hours, and the blue is Debbie, stand for nucleus. You can see some of the protein already got inside of the nucleus, but majority of protein are in the paranuclear space. At 24 hours, now you can see every single nucleus has the protein co-stand with Debbie. So what is the mechanism of this protein delivery? So first we have a QQ modified protein, OCT4, and the QQ modification is so-called non-covalent linkage, does not form any chemistry bond between QQ and the protein. But this non-covalent modified protein can directly penetrate inside of the cell. Once inside of the cell, due to the cell viscosity change, because cellular viscosity is much higher than the protein solution, and then the QQ region dissociate from the protein surface. Therefore, to expose the nuclear localization signal of the protein to directly target the protein into the nucleus. And so that's how the nuclear targeting achieved. If it's a ER protein, then the protein contains an ER localization signal, then it can deliver the protein into the ER, so on and so forth. The protein we have tested start from a small amino acid, 9 amino acid peptide to 230 kilodalton protein. And also from a acidic protein to base protein, we have tried the transcription factor, enzymes, membrane protein, and so on and so forth. So that's the first protein delivery technology platform. The second one is what we call the QQ IPS, protein-induced IPS technology. So as we all know, Dr. Shinya Yamanaka received the Nobel Prize in 2012 because he invented this IPS technique. And so far, this IPS technique has been invented for 16 years, but there's no successful clinical application yet. One problem is the extremely low conversion efficiency, and also associated with this technology, also associated with safety concerns. So here basically explain, the Yamanaka gene IPS technique, and the gene is not a biological active molecule. The gene has to be transcribed into messenger RNA, and then messenger RNA has to move to the ribosome to translate into protein sequence. It is actually the protein which serve as active molecule to convert fibroblast cell into iPS cell. As I already mentioned to you, we have a QQ protein delivery technology. We use bacteria to make the protein and directly QQ modified with the QQ reagent. Then we can deliver the protein into greater than 97% population of the cancer nucleus, so that to generate, to convert the cancer, either cancer cell or fibroblast cell into iPS cell. So this technology actually gives us a greater than 90% of the conversion efficiency from the starting cell into iPS cell, and also without the virus delivery, without the oncogene, because we remove the CMAC and KF4 gene, replace with another protein. And nevertheless, both Yamanaka technology and our technology belongs to what we call the first generation cell reprogram technology, simply because this is an in vitro cell reprogram in the cell culture dish. And currently, all the labs and stem cell company are currently working on this first generation cell reprogram technology. So in order to, QURGEN actually went ahead and invented a third technology platform, which is called QQ protein-induced in situ disease tissue reprogram technology platform. And this is the second generation cell reprogram in situ cell reprogram technology. And here, this is a mice, which contains a tumor. And this is the tumor tissue. So by systemic injection of the reprogrammed protein, and our protein delivery technology can target the protein into the cancer tissue, and to convert not only into the cancer tissue, but also get into the nucleus of the cancer tissue cell to convert the cancer cell into transient iPS cell. And the transient iPS cell secretes exosome, which mimics the stem cell environment. And we call this as a bystander effect. And this bystander effect can convert the surrounding cancer cell into normal tissue cell. And also the in situ generated iPS cell can differentiate into normal tissue cell as well. So this basically performs a cell reprogram in the disease tissue in vivo. And this technology actually has a widely patented internationally. And this is the only safe second generation of in situ disease tissue reprogram technology, which is significantly accelerating the clinical translation of the stem cell technology. And we have data basically shows we can convert in situ cancer cell, many different type of cancer cell and injured heart tissue cell into stem cell in vivo. And here is the application. Using the third technology platform, we actually invent a new cancer therapy called a cell-converting cancer therapy based on a protein drug called the Reprogram. And this slide basically shows on the top is the traditional cancer therapy, regardless of chemotherapy, radiation therapy, immunotherapy, or targeted therapy. All those therapies based on single strategy, which is to kill the cancer cell. When you try to kill the cancer cell, simply because the small chemo drug does not have the targeting capability, which bring a major toxicity and side effect. And also under the selective pressure of the cell killing, and the cancer cell can derive the new genetic mutation, cause the cancer cell resistant to the existing cancer chemo drug. And this is what we call a drug resistant. And if you don't kill 100% of the cancer cell, then cancer will coming back. So therefore, there's no cure for late stage metastatic cancer cell. So our cell converting cancer therapy take a different approach. So simply by intravenous injection of the reprogram drug, anti-cancer drug, the protein can reach to the nucleus of a cancer cell, and regardless is the low grade, high grade cancer stem cell. And therefore, once the reprogram protein reach to the nucleus, will initiate the cancer cell reprogram and to convert the cancer cell into transient stem cell, which will redifferentiate into normal tissue cell. If you continue this reprogram and redifferentiation process, you can convert almost essentially all the cancer cell into normal tissue cell. Not only that, because this is cell reprogram method to treat the cancer, we also rejuvenate the convert a normal tissue cell. So in summary, our new cell converting cancer therapy does not kill any cancer cell at all, but instead it uses reprogram induced in situ cell cancer cell reprogram to convert the cancer cell into normal cell. And this really solved the major problem. We found a minimum toxicity, low drug resistance, and we also, this cancer therapy also achieved very high efficacy. We found, at a very late stage cancer bearing animal, it can achieve a cancer cure, and essentially, and right now we have a phase 1 FDA IND impending for this drug. So the next one is the complete clinical pipeline and the development plan. So in our cancer drug, Reprogram in the US, we're going to focus on 4 cancer indications, including breast cancer, ovarian cancer, pancreatic cancer, and colorectal cancer. But in China, we're going to focus on liver cancer and stomach cancer. And our second pipeline is the anti-heart failure drug, and we call it Reprogram. And the third one is the anti-aging drug. We call the a different drug, protein drug. We call it Reprogram. So we also have a diabetes project and eye disease project. And those are the projection on when the drug can get into the market. So with that being said, so QURGEN invite for partnership. So we're currently seek for partnership for joint development, research collaborations, interesting license in the following area. The first is the drug delivery, protein delivery, utilize our protein delivery technology. The secondly, we are also open for collaboration or co-development on our cancer project, because there are so many other cancer types we have not touched it. We're welcome the opportunity for working with the any partner for the cancer drug. And third one with the IPS, scale up IPS production at the GMP level. And lastly, we are going to advance for revolutionary in regenerative medicine and anti-aging. And so next slide is the proposal of the financing. We're raising currently $35 to $45 million and round just open, and those are were found phase 1A and phase 1B clinical trial, and also partially found a phase 2 clinical trial, and also we for the anti-aging program we want to apply for IND and conduct clinical trial. So this is our QURGEN team, and I'm the chief scientist and also the founder of the company, and those are my partners. And we have Dr. Wang as our CMO, who is a very experienced clinical doctor. And thank you. And finally, I would thank you for your attention.